So in last lecture we study the diffusion. So let's revise what we studied in last lecture. Diffusion. We we studied that there are types that are simple diffusion cell and facilitated diffusion. In simple diffusion cell, a number of apparatus have been described for studying the diffusion of drugs across membranes. A simple cell for the purpose, and the apparatus consists of two cells A and B separated by barrier through which diffusion occurs. The chambers are generally of glass or clear plastic and are easy to assemble and clean. This also allows for the visualization of the liquids and of the rotating stirrers. The chambers can be thermostated and automatic sampling devices can be attached to them. The drug solution is taken in the donor compartment while the solvent is taken in the receptor compartment. Sample solutions are collected periodically from the receptor compartment and analyzed spectrophotometrically. Then next we study in the second part the dissolution. So what is dissolution, and that means what is the definition of dissolution we studied. So the dissolution may be defined as the process by which a solid solute enters into solution when added to an appropriate solvent. Dissolution rate refers to the rate at which the solid dissolves in a solvent. When a solid dosage form such as tablet is put into water, the drug contained in the tablet begins to pass into solution. Simultaneously, the solid matrix of the tablet disintegrates into granules, and these granules in turn disaggregate into fine particles. Dissolution of the drug takes place both from the disintegrated granules and from the fine particles. Although the effectiveness of the tablet is in releasing the drug is somewhat depends on the rate of disintegration and deaggregation, of more importance is the rate of dissolution of the solid drug. Most often the dissolution step is the slowest, and hence the limiting or rate controlling step in the bioabsorption of drugs of low solubility. The rate of dissolution of a solid in a liquid is quantitatively given by the Knowles-Whitney equation. dC by dt is equal to kdA in bracket Cs minus C is equal to dA upon H in bracket Cs minus C, where dC by dt is the rate of dissolution, kd is the dissolution rate constant, d is the diffusion coefficient, Cs is the saturation solubility of the solid, and C is the concentration of solute in the bulk solution. It can be seen that dissolution rate constant Kd is equivalent to the diffusion coefficient divided by thickness of the diffusion layer, that means H. In diffusion theory, it is assumed that there exists an aqueous diffusion layer or tangent liquid film surrounding the surface undergoing dissolution. The thickness of the stationary phase is represented as H. The solute molecules exist in stationary layer in concentration from Cs to C. Diffusion occurs through this stationary layer and mixes in the bulk solution at a distance greater than h. At the interface diffusion layer and the solid surface, the solid solute is in equilibrium when with the solute in the diffusion layer. Then the concentration changes with distance across the diffusion layer. This concentration change, that means the concentration gradient, is constant when C is considerably less than the drug's solubility. Cs, the system is said to exhibit sink condition under these circumstances. The term C may be eliminated from the equation dC by dt is equal to dSCS upon VH. In the derivation of Knowles-Whitney equation, it was assumed that H and S are constants, but actually S decreases as the particle size decreases when the solid undergoes dissolution, and the thickness H of the static diffusion is also altered by agitating. Then next we see the dissolution study, most important. In that, a number of in vitro methods have been described to study the rate of dissolution of granules, tablets and capsules. The most widely used apparatus for the study of dissolution rates of active drugs from solid dosage form as a tablets and capsules are basket and paddle type of dissolution apparatus. The USP basket type apparatus 1 consists of a 40 mesh stainless steel basket attached to the end of the stirring shaft, which is turn is attached to a variable speed motor. A tablet or capsule is placed within the basket and immersed in the dissolution fluid contained in a round bottomed container and rotated at a speed specified. The dissolution fluid is either water, buffer solution or dilute hydrochloric acid, that means HCl. Solutions simulating the gastric pH. The dissolution fluid is maintained at a temperature of 37 degrees Celsius, that means room temperature. During the run, samples are withdrawn at different times for analysis. The volume of the dissolution fluid is maintained constant by replacing the amount of sample removed with fresh dissolution fluid. The analysis of the sample withdrawn at different specified times is undertaken in order to determine the dissolution rate. The USP paddle apparatus 2 consists of a paddle attached to the end of a stirring shaft, which is in turn is attached to a variable speed motor. The paddle is immersed in the dissolution medium contained in a round bottomed 1000 ml container, which in turn is immersed in a constant temperature bath to maintain the temperature at 37 degrees Celsius. During the operations, the tablets is dropped into the dissolution fluid and the fluid is stirred with the paddle positioned vertically near the bottom of the container at the specified speed 52,000 rpm. 52 to 100 rpm. Samples are withdrawn and analyzed as described above. Next we see the Dressman and Repas introduced the concept of using more biorelevant dissolution media. These fluids consist of ingredients that provide physiochemical properties similar to the content of the human gastrointestinal tract. Their composition is given in chart. A practical feature of this physiologically based dissolution testing procedures is that they use compendential devices in combination with the biorelevant dissolution media. The procedures thus provide a strong capability for predicting in vivo performance of the drug and or drug product. More than a more research project, this technology was proven to be useful as a surrogate for bioavailability, bioequivalence studies. The development of dissolution test for less soluble drugs. Less soluble drugs are defined for the purpose of this chapter as those for which the D/D bias is greater than 250 ml at some pH between 1 and 7 even in biorelevant media. However, it would be unwise to simply lump all the less soluble drugs together. Features of the molecules such as lipophilicity, ionization at physiological pH and crystal lattice energy, that means melting point, can all significantly affect the magnitude of the solubility or dissolution problem and the ease with which appropriate dissolution methods can be developed. That said, this section is arranged in substance which reflects the physiochemical properties of the compounds in increasing degree of difficulty from the points of view of developing both formulations for oral delivery and appropriate dissolution test for this formulation. Then next we see the dissolution based biowaivers. The biopharmaceutical classification system BCS takes into account three major factors that governs the rate and extent of drug absorption from infrared, that means IR solid dosage forms, namely dissolution, solubility and intestinal permeability. It classifies the drug substance and therefore the drug products into four classes. The major four classes of bioavailabilities are most important for GPAT. The first is class 1 contain is high solubility and high permeability, and the class 2 has low solubility and high permeability, and class 3 has high solubility and low permeability, and class 4 has low solubility and low permeability. The BCS, that means biopharmaceutical classification system, takes into consideration gastrointestinal physiological factors such as pH, gastric fluid volume, gastric emptying, intestinal transit time, etc., and permeability factors. According to the BCS guidelines, the drug substance is considered highly soluble when the highest dose strength is soluble in 250 ml or less of aqueous media over the pH range 1 to 7.5. The drug substance is considered highly permeable when the extent of drug absorption in humans is determined to be 90% or